Hello everyone, it's Miss Bell, and today we're going to be talking about the scientific process, which is also known as the scientific method. So basically the scientific method is a series of steps used to answer a question or solve a problem. This doesn't have to be a scientific problem, it could be an everyday problem. It could be, why does my dog keep barking at the window? Our little dog Lucy is 15 years old and she's starting to lose her sight. And if we have our window open by where she lays, then she barks. And we figured out that she cannot see. And in addition to that, anytime a dragonfly or a bird goes by that back window, or sometimes even if the wind blows and the trees move a little bit, then she just starts barking. And a lot of the time it's when our child is napping. So this was a problem for us that we had to solve. So that would be an example of something that, you could use the scientific method to figure out a solution to your problem. So anyway, obviously scientists use this, and this being a science class, we're going to talk about it. So the main steps, because it is a series of steps that you see, and every teacher teaches this a little differently, every book teaches it a little bit differently, but these are the primary steps that you'll see. First, you're going to observe something, like the dog barking. Then you're going to define whatever the problem is. Maybe she can't see and she's seeing things fly by the window. And then you're going to propose a hypothesis. Well, let's close the window to where she can't see out the window. And then you gather evidence and test this hypothesis. Well, maybe it's not her vision. Maybe she's hearing things. Maybe she's crazy. Then you test that hypothesis. I would close the window and see if it worked. Well, look at there. It did work. She stopped barking. So I retained my hypothesis, and then I can test this over and over every day to see if I keep the window closed, if she will stop barking, and eventually I could develop a theory if repeatedly the experiment comes out the same way. So this is just an example of how you could apply the scientific method into your daily life. Before we move on to the next slide, I do want to say that this PowerPoint or this video would typically be done over multiple days in class. So we would spend all this time in class and stopping at different points throughout the unit. So this would be done over probably 3 days' time in class. So just be aware of that because this is a lengthy video. So the first step is you've got to find some sort of problem. We typically don't go looking for problems, but it's pretty easy to find them. The question or problem is going to be based on an observation. Observations are going to be typically something you see with your 5 senses. So you might see something, hear something, taste something, smell something, or even notice a touch, some sort of touch. So in this stage, the scientist is going to recognize something has happened, and this is something that continues to happen repeatedly. So one example, and this is a super common example as far as test questions go, is that your neighbor's plants grow better than yours. That could be a problem for you. You want your plants to grow good too. Now, this is what's written in your notes. I'm going to give you another example. I would recommend for you to write this other example down because it's always good to have more information to study rather than less. So this other example is going to be, my lasagna tastes nasty. So this is a problem. I need to fix this. So that brings us to the next step. So my next step is to research. How can I fix my problem, and what is some background that I can find out? So I'm going to explore resources that might have information about that question or that problem. So you could go to the library. We don't typically go to libraries anymore, especially right now. We probably wouldn't go to a library. Now we pretty much use what to research? Google, Bing, the internet, some sort of search engine. We have to be very careful about the sources that we're using to make sure they're a reliable source. Now, another scientist is probably reliable. Some random person that posted something on a blog might not be reliable. You probably want to look at that person's credentials. If you're looking at something medical, then a doctor of some sort, specifically a specialist in that particular area, would be best. If I need a recipe for lasagna, I might look at a famous chef or someone that I know makes good lasagna. So, for example, if I know that my friend's grandmother, let's say my best friend's name is Claire, so let's say Claire's grandmother, if I know Claire's grandmother makes the best lasagna, then she can be my source or one of my sources. I could also look up some recipes. I can go to some famous chefs' websites like Paula Deen or the Pioneer Woman, Ree Drummond, or the Emerald, or you get the picture. So anyway, as far as your neighbor goes, well, your neighbor could be a source. Your neighbor's plants are growing better than yours, then go ask your neighbor, hey, how do you care for your plants, neighbor? And then you might find out that the neighbor is using fertilizer. Well, what type of fertilizer is it? There's lots of different brands, and maybe you're not using it, maybe you are, maybe you need to use a different brand. Anyway, so at whatever point you're going to research. You're not going to just dive right into an experiment. You're going to research. So then you come up to a hypothesis. So through my research, I found out that in making lasagna, I do not use oregano, which is a very common spice that you would find in Italian food. So Claire's grandmother uses oregano and hers tastes really good. So does Paula Deen, so does the Pioneer Woman, so does the Emerald. These are all pretty reliable sources. So I'm going to make a hypothesis that if I add oregano, then my lasagna will taste better. If-then statements are the easiest way to write a hypothesis. Because you're saying, if I do this, then this will happen. You're predicting what will happen. Now, prediction doesn't always come true. The weatherman predicts the weather a lot, but it doesn't necessarily occur. For example, hurricane predictions. Sometimes they're right, sometimes they're off a little bit. So your hypothesis is your possible explanation or a possible answer to your question. And you're going to base that on your research. So you might come up with a hypothesis that fertilizer will make my plants grow faster. So here's a question for you. What is the starting point of scientific inquiry? So scientific inquiry, you can think of that as if I inquire, I'm questioning. So this is a whole questioning process. So you might see that word instead of scientific method or scientific process. So anyway, what do you think is the starting point? So we're looking at the starting point here. So what will happen first? A, theory development. B, observation and question formation. C, experimental design, or D, reviewing what you already know. So remember I said theories don't come until you've done an experiment a bunch of times. So it's not going to be theory development, so we can cross that one out. Experimental design. Experimental design is when you design your experiment. Will you do that before you observe and form a question? No, you can't design an experiment unless you have a question. So you're not going to do that. So I'm kind of going backwards with what you would do. So am I going to observe and form a question, or am I going to review what I already know? Have you ever pulled up the internet, the Google, and you just kind of look at it for a minute like, what was I going to type in? You have to know what to research and review in order to know what to type in your search engine. So, that being said, our answer is observation and question formation. We have to observe a problem and form a question about that problem before I can review what I already know about that problem. Marta wants to conduct a science fair project examining the effects of ultraviolet light on the growth rate of a certain plant species. She will expose 30 plants to high levels of ultraviolet light and 30 plants to normal levels of ultraviolet light. She will compare the average growth rates in each group. So, she's going to look at the effect of ultraviolet light on the growth rate of a certain plant species. That is her experiment. What is the most reasonable hypothesis that could best be tested? Now, a hypothesis must be able to be tested. So keep that in the back of your mind when you're looking at this. It has to be able to be tested with the experiment that Marta has set up. So for some reason, the A, B, C, D didn't come across. So I'm just going to write them in for you. So A, all plant species require ultraviolet light to grow. B, this plant species grows more slowly under high levels of ultraviolet light. C, ultraviolet light has no effect on growth in any plants. Or D, plants require only small amounts of ultraviolet light to grow normally. So the first thing I look at when I look at this is a specific word. We have to be very careful of certain words. One of those words is all. All plant species. Did she test all plant species? She only tested certain ones. That would be a big experiment to test every single plant species. So we're going to eliminate A because it's not going to be able to be tested in this experiment because she only used certain plant species. So B sounds pretty good. This plant species grows more slowly under high levels of ultraviolet light. Did we test high levels of ultraviolet light? Yes, we did. So let's come back to that. We're not going to eliminate it just yet. Ultraviolet light, look at C. Ultraviolet light has no effect on growth in any plants. Again, what word is sticking out to you? Right here, any. Any is saying any plant is basically the same as all plant, right? All plants, sorry. Okay, is she testing any plants? Nope, just certain ones. So we're going to eliminate C. Looking at the last question. So you should be between B and D at this point. We hadn't read D yet. So plants require only small amounts of ultraviolet light to grow. Now, what did she test? She tested high levels and normal levels. Did she test small levels, small amounts? No, she did not. So we're eliminating D, and B is the correct answer. So that brings us to the experiment. So the experiment is the planned procedure to test the hypothesis. So the hypothesis is only going to test one factor or one variable at a time. What that means is you have to split it up into multiple groups if you need to test more than one thing. So the experiment tests your hypothesis. Now, your hypothesis must be testable, okay? So, can I test how many steps it takes to walk to the moon? No, because you can't walk to the moon. So that's not something you can test. So it has to be something you can actually test for it to be a valid hypothesis or experiment. So this brings us to the parts of an experiment. So you should always have a control group and an experimental group for it to be a good, valid experiment. So your fourth part is the experiment. That's why the 4 is right here. Your fourth part is experiment. So anyway, you have your control group. So your control group is a group that gets everything except the experimental treatment. So it's going to get all but one thing. So it will be missing something. It will be missing whatever factor you believe is going to make a change. So constants, the text is messed up a little bit here. So I'm not really sure why that's there. I'm going to just scratch that out. So constants are the same for every group being tested. Another name for a constant is a controlled variable. Do not get a controlled variable mixed up with a control group. Let me explain the difference. So a control group, if we're talking about the plants being fertilized, like we were saying, the neighbor's plants grow better than yours, I'm going to hypothesize that fertilizer will help the plants grow better. Okay, so a good experiment would be to take, let's say, 5 green bean plants and do not put fertilizer on them. Take 5 green bean plants and do put fertilizer on them. So the ones you do not put fertilizer on is going to be your control group. That is 5 plants that have no fertilizer. So 5 plants that have no fertilizer means they did not receive the special treatment, the experimental treatment. They didn't get anything extra. So the control group is not going to get anything extra, or I like to call that extra thing the experimental treatment. So the 5 plants that did get fertilizer is going to go in your experimental group. So they're getting the experimental treatment. They get something extra. So the experimental group, experimental, gets something extra, or you add something to it. Okay, so this is your group being tested. Now, we know what's going to happen with a control group usually, but we need to compare what's normal, the control group, with what's new, the experimental group. Okay, so let's say the plant grows, the experimental group plants grow 10 inches, but the control group plants grow 7 inches. So we need to see that difference. What if both groups grow 7 inches? Is there a difference? Did the fertilizer help? Well, no, it didn't. I need to know that because I don't know how much the plants would have grown without fertilizer. So that's why you use a control group to compare what's normal with what's new. So the constants are in both groups. Remember how we said the control group gets everything but the special treatment, the extra thing? Well, it gets lots of other things though. So both groups we will use 5 plants for. Both groups we will use green beans as the type of plant. I don't want to do green beans for one group and tomatoes for the other group. Both groups are going to use the same amount of soil. They're going to get the same amount of sunlight. They're going to get the same amount of water. They're going to use the same size container. Everything needs to be kept exactly the same except for the fertilizer. If I change more than one thing, then I don't know what affected it. If I water one plant more than the other, one group more than the other, then I don't know if the extra water changed the growth or if the fertilizer changed the growth. So you can only change one thing at a time so that you know for sure that that is what's causing the change. So in an experiment that we talked about, we were testing to see if fertilizer had an effect on plants, specifically plant growth. Now, the other example I was giving you with the lasagna. So my experiment with the lasagna, so I'm going to say my control group, excuse my handwriting, I'm doing the best I can, but I don't generally have good handwriting. My control group will be 5, and they can be mini pans, 5 pans of lasagna without oregano. And my experimental group will be 5 pans of lasagna, with oregano. My constants for both groups. What are some things that's in all lasagna? So what will both pans have? Well, one constant is the number of pans, right? 5 pans. Should the size of the pan be a constant? Or type of pan? A metal pan versus a glass pan. So the type and size of the pan. The temperature it's cooked at. What else? The time we cook it. If I cook one for 20 minutes and one for 50 minutes, that's going to be a big difference. What goes in the lasagna? Okay, your meat, sauce, noodles, cheese, all the cheese. And, you know, whatever else. Everybody makes it differently. So, you get the picture. So, my control group is without my special treatment. My experimental group is with my special treatment. My extra thing. The extra thing is the extra oregano that's being added to the experimental group. My constants, both groups get. Both groups get noodles, both groups get cheese, etc. All right, a social researcher conducted an experiment to test the effect of sleep deprivation on reaction times. So sleep deprivation is being deprived of sleep. You don't get to sleep. So you're looking at the effect of sleep deprivation on reaction times, how fast you react. So 50 working adults in an evening study were awakened every 2 hours during an 8-hour period at night. And then they were asked to catch a tennis ball fired from a machine at exactly 20 miles per hour. So that catching of the tennis ball, that's the reaction time. What would be an effective control for this experiment, which would provide a basis for meaningful comparison? So remember, we talked about a control group as being what's normal and how we can compare what's normal with what's new. The new thing is definitely being awakened every 2 hours and say, hey, catch this tennis ball. I know I would not do well with that because normally I can't catch as it is, but being woken up every 2 hours, that would definitely cause some problems for me. So anyway, looking at our choices A, B, C, and D. A, the same participants' reaction times tested in the middle of the day. B, average reaction times at night for the population at large. C, average reaction times during the day for the population at large. And D, the same participants after a full 8 hours of sleep deprivation. So, what do you think? First thing I want you to look at is you have 2 that say the same participants, and your other 2 choices say population at large. So I want to ask you this question. So what do you think is going to give you more reliable results? If you use the same people or just the general population? So it could be different people every time. So right now the NBA basketball teams are at Disney World. They're playing games or something. I'm not real sure. I don't keep up with it, but I know I've seen it a lot because we like Disney World, and so I see the Disney news. So I'm just wondering if your class would have different reaction times compared to an NBA team's reaction times. So should you do the same people every time, or just randomly do these people one time and then randomly do these people another time? So it's definitely going to be better to do the same participants. So we can eliminate B and C because of that. Okay? So we have either the same participants' reaction times tested in the middle of the day, and then the same participants tested after a full 8 hours of sleep deprivation. So if you're looking for a control group, your experimental group was up here, right? They're sleep deprived. You're waking them up every 2 hours. So here's your experimental group up here. Do we need another experimental group? This will be another experimental group testing sleep deprivation. So what is most normal would be when you're not sleep deprived, when you're awake. If you're tested in the middle of the day, you are awake. That is going to be more normal results. So that would be a better comparison, a better control group. So this one was kind of tough. Which of these is kept the same for all groups being tested in an experiment? So it's the same for all groups. Would that be the conclusion, constant, hypothesis, or variable? So this is the definition of a constant. Remember, a constant is also known as a controlled variable. But the variable wouldn't be right because the independent variable, we hadn't got to this yet, independent variable might be different for each group. It's not the same for every group. That's one of the things that is different. That's actually the extra thing the experimental group gets. And we're fixing to talk about that. The questions that we're doing, you might see on your test. So that's why we're going over these questions, just in case you see them again on your test. In fact, several of these will be on your test, but not all of them. A social researcher conducted an experiment to test the effect of sleep deprivation on reaction times. 50 working adults in an evening study were awakened every 2 hours during an 8-hour period at night and were asked to catch a tennis ball fired from a machine at exactly 20 miles per hour. So same situation as we talked about before. So which of the following would not be an affected added control? So what is something we could add to which would not be good to add to this experiment to make sure that we keep the experiment in line and controlled? Would we want to have participants' diets restricted? Participants' sleep patterns recorded for 1 month prior to the experiment? Would you want participants to select wake-up alarms? Or D, the population of participants is increased. So what would be not good here? What would be not good? So I'd like to go through and look at what is good. Let's start with A. Do I want to restrict someone's diet and make them sleep deprived? I would not be a happy camper if that was the case. The problem there is that you're changing two things. So how do you know what affected the reaction times? Was it the sleep deprivation or was the restricted diet? Would this be good? That's a hard no. So this would not be good. Okay, so let's go back and look at our other choices. Even though it looks like A is a good answer, might not be the best answer. Let's double check. B, participants' sleep patterns are recorded for 1 month prior to the experiment. So this means you're just going to write down your sleep patterns for a month. That way you can kind of see what's normal. So remember, the control is what's normal. So it would be good to write down your sleep patterns. If you have a newborn baby, your sleep patterns, it might be normal for you to wake up every 2 hours. That's important. That person might need to be excluded from the study. So this would be a good thing to do. Wake-up alarms are selected by each participant. How do you wake up in the morning? Some people's parents wake them up. Some people wake up because the sunlight comes in. Some people have an alarm that's real soft. Some people have a louder alarm. Some people snooze and they have to hit the alarm, snooze over and over. Either way, if the participant selects their wake-up alarm, that keeps it consistent with how they're used to waking up. If you change the wake-up alarm and you change their sleep deprivation, that changes more than one thing. So it would be good if they selected the same wake-up alarm that they normally do. So that is good. Population of participants is increased. So this would be like saying if I had 10 people in the study originally, you increase it to 50. That is good. That is always a positive thing, and let me tell you why. Normally I use the flu as an example, but right now I'm going to use COVID-19, the coronavirus, as an example, because why not? So if you have an experiment with 10 people in it, and one person gets the Rona, COVID-19, that's going to mess up 10%. 1 out of 10, that is 10% of your results that will now be messed up because that person is sick. The reaction times will probably be affected because of the sickness. So really, that makes that invalid. That person really shouldn't even be included in the study. Well, if you increase that to 100, I'm going to use simple math for you. If you increase it to 100 people in your study and one person gets COVID-19, then only 1% of your results become skewed, become messed up. So only 1% of your results will be messed up. And that's why it's good to have more people or more participants. So you increase the participants. That's the same thing as saying you increase the population of participants. That is always a positive change. Decreasing is a negative. A scientist wanted to test the effect of bright sunlight on cave grasshoppers. Cave grasshoppers are accustomed to total darkness. They live in a cave. During the experiment, what is the purpose of keeping some grasshoppers in total darkness? Why are some of those cave grasshoppers kept in total darkness? Is it A, they would serve as a control group? B, they would exhibit the effects of bright sunlight on cave grasshoppers? C, they would serve as the test group? Or D, they would be subjected to later studies? Okay, so basically you have two groups. You got cave grasshoppers, and some of them are going to be in the sun, and some of them are going to be in the cave. What's normal for the cave grasshoppers? The cave. So this is what's normal. This is what's new. The sun is new. So what did I say the normal group was? Control group. So we keep them in the cave for our control group. We put the ones in the sun as our experimental group. So I can compare the two. I need to compare. Maybe they're going to look at the movement of them. Let's say they're going to look at the movement of them. I need to know how much they normally move. So I might compare how much they move in the cave versus how much they move in the sunlight. So they would serve as the control group. So the answer is A. Okay, if they're in a cave, they would not exhibit the effects of bright sunlight because they're not in the sun. It asks, what is the purpose of keeping some in total darkness? They would not serve as a test group because that would be the ones in the sun. And they would be subjected to later studies. That was just to give you another choice. It's a dummy choice, really not involved. All right, a placebo is inert. Inert means inactive. It does nothing. It's basically like a sugar pill. You can think of it as a sugar pill or a fake pill. Because sometimes sugar might do something to you. Anyway, so it is always given to the control group. The placebo is always given to the control group. You ever have a scenario and it asks you about a placebo, which group it goes in, it is always the control group. Or if it asks you to identify the control group, it is the one with the placebo. And it will have no effect because normally we don't take medicine. And when you think about what's normal, think about how nature is intended, okay? If you think about animals living in the wild, think of if humans were to live in the wild, if it's an experiment with humans. So think about how nature exists normally without, intervention. So anyway, a placebo will have no effect on the control group, so you can compare what's normal in the control group with what's new, whatever is in your experiment, what you're testing. The reason why we use a placebo is to eliminate bias. Remember, bias is always bad. Okay, bias is your opinions influencing results. We're going to talk about that a little bit more. Anyway, placebos are often given in experiments involving people. Humans are awesome because we have the higher order thinking that a lot of other animals don't. But because of that, we can be biased for many reasons. So the bias is your attitude or your opinion factoring into the experiment and affecting the results. So we use placebos to eliminate that bias. So here's an example for you. This is very common for humans. A pharmaceutical company may want to test the effects of a blood pressure medication. The experimental group will receive a pill with the medication. The control group would receive a placebo, a pill that does not contain the medication. The group members will not know if they received the pill with or without the medication. So people who think they have been given the medication, even if it is a placebo, can sometimes improve a health condition just because they believe that the medicine they receive will help them. This is called the placebo effect. Okay, so in this case, it's blood pressure. So they've done tests, lots and lots of tests, blood pressure pills, other pills, like maybe it's like a mood stabilizer pill or like anxiety medicine or something, lots of different medicines. And they always use a placebo now because they realize the placebo effect. So just pretend that you have a test, you have 20 people. Let's say you have 20 people, and 10 people get the placebo, 10 people don't. You don't know which 10, but the doctor's going to come in, and in the best experiment, the person who is interacting with the people in the study are not going to know what you get. So whoever's talking to you, who's ever doing tests on you, they should not know which pill you got. A separate person should keep that record. So anyway, they might say, all right, person number 3, how do you feel today? How's your blood pressure been? And then they might check their blood pressure, and they'll know that you're doing a test on blood pressure. So if you come in every day and you're testing blood pressure, you might have lower blood pressure just because you think you're getting the medicine. Same thing with a happy pill. If I say, hey, do you feel any happier today? Well, if I ask you that enough, you might just start saying yes. So that would be the placebo effect. So the best way to do these kind of tests is when the doctors do not know which participants have the placebo and have the regular experimental drug. So this keeps the doctors or the researchers from treating the participant any differently. Are you sure you don't feel any happier today? So this is called a double-blind study, and that eliminates bias. So a double-blind study is going to eliminate bias. That's going to prevent your results from being messed up by attitudes and opinions of people. Because if you don't know what you're getting, then you're not going to influence the participant. So let me give you an example of this. So I'm going to go back to COVID-19. So right now, COVID-19 is, a big deal, and they are developing vaccines for it. Multiple vaccines are in development for COVID-19. So, if I am developing a vaccine for COVID-19, I have a large stake because if my vaccine works, I will make a lot of money. A lot of money. So, I do not need to be the one performing the study. If I'm the one that created the vaccine, I really don't need to be the one performing the study because someone can come back and say, oh, you have something to gain, so your study's not valid. Maybe it is a valid study. You got to be able to prove it, though, and a double-blind study is going to help you prove it. So it kind of protects everyone because if I want my vaccine to work, I might say, are you experiencing any symptoms? Oh, you're not? Oh, you had a headache? Oh, it was because you looked at the sun too long, right? Okay, we're just not going to put that down. Oh, you had a weird rash come up? Oh, that's just a sun rash. Because didn't you say you used a new sunscreen? Yeah, that's all it is. You're allergic to that sunscreen. It's not my vaccine. Does that make sense? I'm saying that like you can respond to me. Okay. In an experiment to test their new drug on humans, 2 scientists planned to give volunteers one of 2 drugs. Drug A, the experimental drug, or drug B, the placebo, the sugar pill. Which of these would most effectively reduce the effect of bias of the scientist who developed the experimental drug? Do I want to A, introduce a 3rd drug into the experiment? B, decrease the number of experimentals from 2 to 1? C, have a 3rd researcher label and distribute the samples? Or D, replace a portion of placebos with aspirin? First off, I want to come up here to drug A and drug B. Which one is your control group? Remember, placebo is always in the control group. And we have to have a control group for it to be a good experiment. So that means the experimental drug goes in the experimental group. Good experiment with, what little information they gave us. All right, so we're looking at what's going to reduce bias. So placebos do reduce bias. That's great. Introducing a 3rd drug into the experiment, is that going to reduce any bias or opinions? Remember, bias is like opinions of experimenters. No, adding an additional drug is not going to do anything to that. Decreasing the experimenters from 2 to 1. So you're taking 2 people and decreasing it to 1. That's definitely going to be very bad. It's not going to reduce bias. It's the opposite of what you would want to do. Have a 3rd researcher label and distribute or hand out the samples. That's definitely going to reduce bias because that 3rd researcher labeling them and giving them out, they're not going to have stake in the results. Okay, they won't be the one to take symptoms. So they're the only one that knows who's getting what. And so the person that would come in and ask their symptoms and whatnot, they can't influence because they don't know who got what. Replacing a portion of the placebos with aspirin. Aspirin is a drug, so that would eliminate your control group, which would be very bad. That would not help bias in any way of the scientist. So C is your best answer. All right, that brings us to independent and dependent variable. And we've already kind of talked about these, but just not in specific words. So first off, the x-axis, so if you're looking at a graph, the x-axis is the independent variable, and this says the y-axis is the dependent variable. Okay, so you need to know the x-axis is always going to be plotted this way. The independent variable will always be plotted horizontally on the x-axis, and the dependent variable will always be plotted vertically on the y-axis. All right, so the independent variable is your variable being tested. This is what changes in the experiment. Okay, and like I said, it's on the x-axis of a graph. Okay, so this is the extra thing, or what's added to the experimental group. This is also, the independent variable is the only difference between the control group and the experimental group. That should be the only difference between the two groups. So the independent variable is just the variable. Okay, so in the case where we were talking about earlier, where we were going to test if fertilizer makes my plants grow better, my independent variable, what's different between the two groups, what I add to my experimental group was the fertilizer. Okay, now notice I didn't say the 5 plants that received fertilizer, because the 5 plants that received the fertilizer was the group, because it was 5 of them. So they're in a group. That was the experimental group. The fertilizer is what they received, so that is the variable. That is the independent variable. Does that make sense? Here's another example. Lasagna. If I add oregano to my lasagna, then it will grow better. What did I add? What is the extra thing? Okay, it is the oregano. So in the lasagna, oregano is my independent variable, and that should be the only difference between my two groups. Notice I said oregano is my independent variable, not the 5 pans of lasagna that got oregano, because the 5 pans of lasagna that got oregano was my experimental group. Now, the dependent variable, this is where people tend to get messed up. They want to make the dependent variable the control group. That is not the case. The dependent variable is what you measure at the end of the experiment. So at the end of the experiment, I'm going to measure the growth of the plants. Usually, you also might say height. I didn't spell that right. I'm a little tired. Anyway, so the dependent variable in the plant's case, what I'm going to measure is the height or the growth. I'm going to take a ruler or a meter stick and see how tall each plant grew. And then I can make a determination of if the fertilizer helped them grow better than my control group. So that would be my dependent variable. My height depends on my fertilizer. If I add fertilizer, then plants will grow better. My plants will grow better. So my height, my dependent variable, depends on my independent variable, my fertilizer. So in the lasagna, I'm going to measure, now this is weird, I'm going to measure the taste. Now remember I said you're going to observe with your 5 senses. So taste is hard to assign a number to. So you would say, okay, this one tastes better, or you could rank them from like 1 to 5, comparing them. So you don't always have a quantitative measurement. Okay, so like if your height was 5 centimeters versus 10 centimeters in your control group versus your experimental group, this would be quantitative data. Because it's a quantity. N is for numbers. Anytime you see some kind of number like that taken at the end of the experiment, that's your dependent variable. It grew 5 centimeters. That's your dependent variable. That's what I measured. I measured that number. Okay? So the taste of lasagna, that's going to be qualitative data. Okay, so you're looking at the quality of something. So that's going to be more like using your words for a description. So, independent variable is what's added, what's changed, what's extra, what's different between the groups. The experimental group gets the independent. The control group does not. The dependent variable is what I will measure at the end of the experiment. It's going to help give my results. So, drug manufacturers must test their new products before they can be marketed. A pill containing a new drug was given to Group A, and a pill containing no drug was given to the same number of participants in Group B. In this test, the group given to participants in Group A, so that's this one, the new drug was so the drug, was it a conclusion, a hypothesis, the dependent variable, or independent variable? So the drug can't be the conclusion or the hypothesis. So you should be between independent and dependent. Okay, the new drug is what's added. It's what's different between the two groups. So what did I say that was? Should be independent. Okay, remember independent variables, what's added, what's extra, or what's different between the two groups. So what's added, what's extra to the experimental group, or what's different between the control group and experimental group. Remember, the dependent variable is what you measure at the end. So you're going to measure the effect of whatever that drug was. So your dependent variable is going to be the effect of the drug. Which of these is different for all groups being tested in the experiment? So it's different for all groups being tested in the experiment. It's a hypothesis, constant, control, or variable. So your hypothesis is going to be the same for your experiment. Your constants, remember the definition of a constant is things that are the same for all the groups. So if it said which of these is the same for all the groups, that will be constant, but it doesn't. Control, is your control group in every group? No. If it meant controlled variable, that's a constant, so it will be the same for all groups. So it's not control, so it is going to be your variable. Your independent variable is going to be different for all groups. Let me give you a scenario. Back to the lasagna, okay? So I could make 5 pans, and we'll do different groups. So the first group we'll say is our control group. So that's my 5 pans without spices. Experimental group 1, we're going to add oregano. Experimental group 2, let's say with basil. Okay, so separate pan of lasagna with basil in it. Experimental group 3 with garlic. Does that make sense? So oregano is an independent variable. Basil is an independent variable. Garlic is an independent variable. So you can test more than one variable, you just have to put them in different groups. So that would be an example of that. Data. We need data for an experiment. So data from the experiment is collected, written down, which is recorded, and analyzed. So you're going to analyze the data to see what your results are. So you might have written down the differences in growth of your plants. So like I said, if we did 5 plants, then I need to write down how many centimeters each plant grew, because I'm not going to remember that. And typically you would measure them on multiple days, not just one day. Then you have your conclusion. Your conclusion is going to state whether your hypothesis was right or wrong, correct or incorrect. You're going to either accept or reject your hypothesis. If your hypothesis is correct, good job. Your problem is solved, hopefully. If it was incorrect, then the experiment failed. That's okay. Make a new experiment. Try something different. Maybe the oregano didn't really help. Maybe I didn't add enough oregano. Maybe I need to add oregano and garlic. Okay, so you can try different things to see what you need to look at, to change in order to make the result what you want it to be. So, example is my experiment showed plants with fertilizer grew faster and bigger. So we did note that the plants grew better with the fertilizer. With the lasagna, like I said, it may have tasted better, it may not have. It could go either way. So it's okay if your experiment fails. Use the data that you received from your experiment and build on it to make a new experiment. Famous saying, took 200 times to get the light bulb right. Something along those lines. I'm sure I messed it up. But the point is that don't expect the first time to go perfectly. I know I'll use cooking as an example because, most people cook, especially if you live in Vancleave because there's very few restaurants. I use cooking as an example because we can look at all the ingredients and we can change things. Just cooking something for 1 minute longer can change the taste and the texture. So we can change a lot of variables with cooking. And you might think it was good when you cooked it that first time, but if you make more changes, you can make it even better. So, for a conclusion to be meaningful or valid, the experiment must be repeatable with the same results. I should be able to perform the same exact experiment, Okay, so with the plants, I should be able to take the same exact plants, use the same amount of water, sunlight, all that jazz, same amount of fertilizer, and reproduce the same results. If I do not, then my conclusion might not be valid or meaningful. This means in your experiment, you must write down detailed instructions. So when you get to your scientific method in an experiment, when you start using the scientific method in an experiment, it's very important when you're writing your experimental design and procedures to be detailed. I used 20 milliliters of water, or they were kept at a temperature of 32 degrees Celsius, or just be detailed. Because you need to be able to repeat it for the conclusion to be meaningful and valid. Now, graphs, models, diagrams are very good to illustrate your conclusion in a visual manner. Would you rather read 3 pages of written paragraphs, or look at one chart or graph and see, okay, this column is higher than the other one. Well, my experimental group must have grown better. It's just easier to look at graphs, so we like graphs. So a theory. A theory is a set of related hypotheses. Hypotheses is plural for hypothesis. That have been tested and confirmed many times by many scientists. Okay, so if you're the only one performing the experiment, then it's good to get a peer review. So that's like another scientist. Peer review is very good to find any inconsistencies and also to provide support for theories. So anyway, many times by many scientists. Okay, this is repeated. So this is repeated experimentation. A theory is going to unite and explain a broad range of observations. It's not the same as being general, and it never implies a lack of knowledge or even a guess. Remember, a hypothesis is a guess. Theory, you have knowledge because you've proven it multiple times. One example of a theory, which we'll be getting into immediately following this, is the cell theory. The cell theory basically states that the basic unit of life is cells, and all living things are made of cells. So this is something that we've known, for a long time, for more than 100 years. So this is very commonly accepted knowledge, but back when it was first discovered, it was not commonly accepted knowledge. And so several scientists got together to formulate the cell theory, and then other scientists have continued to add to the cell theory to further support it based on repeated experiments. So we're going to talk more about that and how that relates to theories.